It is our very great pleasure to introduce Professor Sheila Patek from Duke University. Dr. Patek is the Hehmeyer Professor of Biology and a Bass Fellow at Duke. Her lab is interested in using multidisciplinary tools to find the unifying principles that guide, limit, and promote the evolutionary diversity of biomechanical systems, primarily through studying the fastest biological movements on the planet. Some examples of the fascinating systems that she studies are the ultrafast movements of mantis shrimp and trapjaw ants, and acoustic communication in spiny lobsters and mantis shrimp. Some of you might have already had an opportunity to hear Dr. Patek speak on NPR, CBC, BBC, or in her fantastic TED Talk from a few years ago. So it's a great honor to have her presenting in our Mathematical Laws of Morphology and Biomechanics series. So without further ado, please take it away, Dr. Patek.
All right. Well, thank you, both of you, for the invitation and the opportunity today. The title of the series, Mathematical Laws of Morphology and Biomechanics, when I saw that, I was like, whatever, I'm there in whatever form it takes. It's an incredibly interesting area of research and an area really worth discussing. But I also think it's a moment in sort of the history of biomechanics and morphology when we have an opportunity to really revisit some of the ways that we think about these systems. And it certainly, I've been feeling that way about my research program, which felt at the beginning just, completely about discovery. And now I feel like we're at a cusp of pulling together those discoveries into something that might be considered a rule or a law or some, broad framework. And so that's what I'm going to talk about today. And let me do that now. So all this practicing was so worthwhile of learning how to use WebEx again. Okay. So today I'm going to talk about small, springy, and ultra-fast organisms operating at biomechanical and morphological limits. And what I'm going to do today is start in a space to develop some intuition where in some ways intuition doesn't really work in these systems, but at least to get us on the same page, but really get us towards thinking about a class of systems defined by their size and their morphology and their mechanics. And by the end of the talk, I'm hoping that you'll be able to join me in thinking about a biology-wide transition between ways of developing movement. So what I'm going to do is attack our intuitions and lack thereof with a few examples, and I'll be integrating a couple of themes here. And one of them is energy. When we talk about small, in the systems that I'm going to talk about today and focus of my lab presently, small means that they don't need much energy to run. And this is a crucial piece to understand about these systems, because a lot of people talk about like the most sort of energetic, powerful movements ever. But actually, what we're talking about is not the amount of energy, we're talking about the rate of energy, the rate at which energy is released. In other words, it's a small amount of energy, and then you divide that by time, and then you have a really high rate. So we're talking about high rate. And then ultimately, when we talk about morphology and biomechanics, we're talking about how these organisms control the flow of energy through the system. So that'll be one theme that you'll see, and we'll gradually ramp it up to some pretty heavy hitter ideas at the end. And that's how I think about this. Amount of energy is small. They're small organisms. The rate of energy has a lot to do with the fact that they're springy. They use springs to reduce the time it takes to use that energy. And the flow relates to how fast all of this is happening and how it's moving through the system. And that's why we say ultra fast. And then the third theme, because I always like to have lots of themes, is about whole organisms, because I felt like that was a big theme of this series. Limits, obviously, if we're talking about laws can often be limits. And then ultimately, and at the end, how if we look at organisms and their limits, we immediately find ourselves in a pretty amazing interdisciplinary space, especially if you take a quantitative mathematical approach that can be applied to any system. So hopefully, Alessandro and Kathleen are feeling happy now, because I think that's what you guys are talking about. And if it's not, let me know afterwards. All right, so let's start with organisms, and I want to move this little thingy out of the way. There we go. And I wanted to start with just kind of a nod to where many of us have been for the last couple of years. Normally, in my career, I have spent all of my life just on planet science because I feel happy there, and it's easy to be there. And then this pandemic more or less threw me off of planet science, gazing back, wishing I were back on planet science. So I'm going to start today by, if you, just on the off chance you are not presently on planet science, to invite you there by talking about some organisms that are small, springy, and ultra fast, and also, just as an aside, to get you interested and to develop some intuition about what we're talking about in terms of these organisms. So our first example is going to be our gall midge larvae. This is a system that it was actually a really awesome collaboration by many members of my lab. And what you can see in this picture, and hopefully you can see that little like laser pointer thingy. It's very cool. You can see right there, there's like a little orange thing. That is a gall midge larva. So when I say small, I mean small. So anything energetically that this animal is going to do is going to require a small amount of energy. However, this animal can jump with just one jump from here to the tip of this pen. So we're talking about quite a few centimeters. And it does this without legs. So now we're starting to talk about the how this animal could possibly control energy in a way to allow this to happen. So this is a concatenated video. I was actually gearing up for some really awesome Winter Olympics watching this weekend, and I noticed in the New York Times today they had like one of these composite pictures of one of the skiers spinning around. I was like, oh, what do you know? So that's sort of the same idea here, but the difference here is looking at the space of this thing compared to Olympic skier. This is 5mm scale bar, and this is up to a jump that takes 100 milliseconds. So small and yet brief. So when we want to connect that to how does this animal do this thing, we started to talk about rate and how does the animal actually control this. And what we discovered is that they essentially flex their body, and I'll show you in a second. They attach their head and their butt together with some adhesive hairs, and then they store elastic energy in their body. And then once that latch releases, it allows them to essentially push off with part of their wormy body that is essentially kind of working like a temporary leg. So their control of the rate relates to storing up energy ahead of time in its springy body and then releasing it suddenly. So small energy, high rate. And then the flow is largely controlled through a latch. So if you look carefully here at the start of this video, you can see a little protuberance sticking out, and it's essentially sticking out and touching the other side. And once that releases, the animal starts to jump. So this is essentially what it is in a still. They form a loop. Some kind of hinge shows up, some weird morphology we don't understand. They contract this to somehow store elastic energy in their flexy body. And then they position themselves to launch. And then this little latch right here releases. And that hinge that we don't know where that comes from allows them to push off with this one leg, and then they take off. So this is a simple example of amount, rate, and flow. And for our morphology buffs today, what's really cool about this is this discovery is that they're essentially using these microscopic hairs. Glad Travis is here today because he knows what I'm talking about. They're kind of like gecko hairs that essentially stick together through just tiny material processes that and then at its threshold release and allow the jump to happen. But what's pretty rad is that actually lots of insect larvae have little hairy patches, and so does the larvae, this larva, all along their body. They use it for locomotion and moving around. But this animal has evolved to use 2 patches of it as a latch for jumping, which is just cool. Finally, This is a picture of where they're found in the wild. They're little larvae and they hatch and they come out of these galls and they fall to the ground and hop around. Okay, that was example one. And if you're really bored and not inspired by biology at this point, you should just sign off. So funny saying that on a Zoom. Sometimes I wonder if I said that in my physiology class, how many students would get up and leave. No, they wouldn't do that. But on a serious note, hopefully that gives you just kind of an intuition. Thank you, Alessandra. You got to have humor here in virtual land. So hopefully that just gives you a gentle lean into this space, maybe just launched you onto planet science to be a little curious about what could be going on in these systems, but also to start to think about their origins, evolution, and diversity, and the principles that allow them to do this awesome stuff with small, springy, and ultra-fast amount, rate, and flow. So let's talk about limits for quite a few minutes here. And we'll start by talking about another recent discovery in my lab. Again, a really nice collaboration of many lab members. And this is a story about a crustacean called an amphipod. And they're really small. So check out the scale bar, that's 2 millimeters. And the males, have this huge snapping claw. And the dactyl of the claw fits through the eye of a needle, or since you all mostly don't have your videos on, you can do this. You can take hair if you're not bald or you have some. The dactyl is the diameter of a hair. Okay. When we say small, that's small. Okay. And they're found actually right off the docks of the Duke Marine Lab. And that's my team out there finding them. So again, amount of energy has to be small because the animal's small. So what are they doing? They have essentially figured out how to develop extremely fast motion with that little hair-like diameter of the claw. That's a real-time video from some colleagues in Japan who introduced us to this system. And let's talk about what we mean by rate and fast. So this video was filmed at 300,000 frames per second. And I was mentioning at the start of this thing, I was up at Brown recently, Brown University, giving a seminar. And I asked that group, because there's a lot of biomechanists there, like, what's the fastest frame rate? All those fancy folks who study bat locomotion and things like that film at. And the highest number that anyone said was 1,000 frames per second. So when we talk about limits and ultra fast, 300,000 frames per second, it doesn't even register basically for most biomechanics research. Even the fast stuff doesn't capture this. So it's basically impossible for us to think through what this motion is. It's just like completely out of the realm of understanding. And one pathway to thinking about it is through comparisons of acceleration. So this is the strikes that we just looked at are amphipod strikes up here in orange. And we have some other super fast animals like trapjaw ants, which we're going to hear about in a few minutes. But essentially, these animals that we report in the Current Biology article turn out to be just at the left-hand most size in terms of mass and extremely high acceleration that have ever been recorded. We have a much more completed graph at this point from Jacob Harrison's dissertation, which you will see soon, no doubt. But essentially what you see when you fill out this space is that there's like an empty space here on the top left-hand corner of this graph. And in this space are other ultra-fast animals and organisms that break when they make motion this big. But we cannot find, or we have not yet found, anything that is smaller and this fast that doesn't break. So this appears to be a limit to self-destruction during ultra-fast movement. Now, some of you are looking at this and like, give me a break. There's not enough data on this to make that point. I'm going to come back to this. I'm kind of setting up something we're going to talk about in trapjaw ants about why there might be a limit. And it relates to energy and springs and how much energy you can put in a spring. And if you're going to use a spring to repeatedly generate motion? How do you do that? And what limits do you hit? So we'll come back to that. But the other thing that we will talk about a little bit later is this line, which I'll show you a much more populated one, and I'll show you an even more interdisciplinary space, which is essentially the other direction, which is that the bigger things get, the smaller they go. And this whole business of being able to make movements more than once means that you got the bullet over here, which is a self-destructive type of motion. These organisms are able to move at the acceleration of a bullet in a gun over and over again. So there's a capability here that relates to morphology and size and biomechanics that allow these organisms actually at this point to still circumvent limits that engineered systems are hitting at a pretty challenging location. You pretty much can't get much smaller and faster so far in engineering. However, by the end of the class, I just finished teaching physiology. At the end of this lecture today, I will start to explain how we might actually be able to get engineering capabilities closer to organisms by understanding the source of these limits. Okay, so now we're going to talk about some math. And I'm not going to do any equations today. So this is about how do we get to the point of setting up a problem that we have established, as I sort of did by example today, through discovery of biological systems, operating at limits, identifying the key parameters. And one of the crucial junctures in our research was the realization of a hard transition and limit that applies to physics, biology, and engineering. Obviously, if it applies to physics, it applies to biology and engineering. And this is when you want to, or when it is effective to generate a movement with stored elastic energy versus a traditional motor, like literally a motor in engineering, or for example, a muscle. And what we found was when we developed just a really simple physics-based math model where we basically propelled a little projectile, this little purple jobby here, and we propelled that projectile with either a motor pushing it, or we used that motor to put energy into a spring and then let the spring push the projectile. Of course, this is mathematically we did this. And what we found is that basically, if you're going to use a motor, it works okay at any mass. But if you switch over to putting the motor's energy into a spring-like structure, you get tremendous effect of the power output of the system. This is mechanical power, so energy per time. That's our rate business here. So tiny amount of energy, very high rate, by basically making the time small. Not only that, but previously people had talked about this from the perspective of ideal springs, Hookean springs, springs with linear force relations and potentially no losses, or at least linear behavior. But we just basically gave these springs a little bit of mass, because it turns out at these small sizes, inertia matters to these organisms. In fact, Hookean assumptions are not correct at these small sizes. Some of you might be looking at this graph, if you love these kinds of graphs, and saying, well, goodness, okay, I get it. At below a certain mass, spring improves power. But why would you want to consider a real spring and latch in your simulation if it looks like the behavior actually is not as good? And the reason is a pretty major one. Okay, so I just want to reiterate, this is just a physics rule that had been thought about previously, but we formalized it here and built on it tremendously in this paper. But essentially, this is a major transition defined by whether you directly actuate something with a motor, or do you put energy in another structure with that motor and let that stored energy develop the motion? And as you'll see as I go, we are now testing for the presence of this transition at various levels in biology. But I want to come back just for a second to the ideal versus real. And it gets to something that's incredibly important here. Along the way in my career, some of you have known me this long, most of you have not. Well, actually, Kathleen mentioned this at the beginning. So at the beginning of all this, right, back 20 years ago when I started working on the system, the whole deal was this. We had discovered the fastest movements on the planet. And my entire career was all about, I felt like, to the public facing, not the actual research in my lab or for fellow academics, was sort of like, did we find the fastest, or is it even faster, or how does it compare to a bullet, and da-da-da-da-da, which, you know, is fun for a little while. Well, I mean, I think it's fun, it continues to be fun for the universe, but like for me, as an intellectual, it's not quite as fun because you got to ask yourself, why are they moving so fast? And do they always want to move so fast? If you give a spring real properties, it means something really much more valuable. It means that you can actually get different performance out of these systems. And if you can get different performance out of these systems, it means that you would want to use them for jumping versus puncturing versus striking versus whatevering. So what this graph shows you is that when we change the y-axis to takeoff velocity, takeoff duration, power delivered to the projectile, maximum kinetic energy, you can see there are times when you would want to use a spring and not want to use a motor. Other times where they're more or less equivocal. And the mass transition shifts. So I'm sorry if I'm going on a big geek out, but it's not every day I get to be invited to give a seminar that is like literally supposed to be about this, because usually this is too heavy duty for a sort of a general biology audience. But my point is that by incorporating these realistic parameters, we suddenly get much closer to the diversity of uses in biology and also detect what might be a pretty profound limit for any of these systems, and that could explain some of the strategies that we see through evolution and morphology. Okay. As we were working through this process, we realized we needed to actually name these systems. So we actually named this class of systems, and we call them latch-mediated spring actuation. And so in short, it's LAMSA. And the reason why we did this is that we realized that so much previous work for 50 years on organisms had just focused on just the rate part. How much energy can you get out per unit time? But really the magic in these systems is all three of these, most especially control of energy flow. So if you think about it from that perspective and a morphological perspective, what these systems are really made of are a motor, and of course this is exactly how archers do this as well, although biology does it much better than archers, which I'll talk about in a second. You have a muscle, that motor, that puts energy into the spring, which is the deflecting bow, and your fingers here are serving as the latch. And then once you release your fingers on this latch, the energy in the bow fires the arrow. So we have a muscle, a spring, and a latch. This is spring actuated, and the energy release is latch mediated. There's a lot of magic in here. So if you gave this person with exactly the same arm muscle a spear to throw, or you gave this person that arrow to throw, it might do well with the spear because of those transitions in mass and when motors are good. But you would not be able to throw that arrow at the same speed as you can when you put the energy from the muscle into a spring and take the mass of your arm out of the arrow. These very principles are what we find when we look at the morphology and the mechanics and the control of energy flow in these organisms. That's why we named it latch-mediated spring actuation, in order to incorporate all of this. Okay. So let's go to our friends the Mantis Shrimp and explore this a little bit. This is the system that I began all this work on. Mantis Shrimp are these like really awesome crustaceans that have a diversity of appendages that they use for different uses. One is what's called the smasher, that has a hammer, and they use the hammer to break open hard-shelled prey. Just to give you again a sense of amount and rate, this is filmed at 30,000 frames per second. Each one of these hammers is the mass of 2 toothpicks, and they are moving at the acceleration of a bullet in a gun. So small energy, it is very small, and the magic is the rate. And the rate, as we've already established, is from springs and latches. So let's look at the morphology of this. So these animals have extensor muscles that contract to basically open the appendage and move that hammer, okay? And the animal's perfectly capable of moving the hammers around directly with the muscles and never using any springs. So they pick up stuff, carry things, do whatever, just like we do with our muscles. But they can shift the whole mechanism over to serve as a lambda mechanism by simultaneously contracting extensor and flexor. So it's one wants to open it, but one is keeping it closed. The extensor wants to open it, but the flexor, try to see how I make this it's visible in the video. The flexor is keeping it closed. Once that happens, the extensor can load elastic energy into deformation of the exoskeleton. And when it's ready to release, the flexor muscles can relax and the system can fire. So here we have that transition business that we were talking about a muscle putting energy into a material and then letting the material actuate the motion at a much, much, much shorter time period in order to enhance the mechanical power output. Just as an aside, because I know I'm amongst morphologists here, one of the really cool things about the system is the latch. And we've done a lot of work on this recently that I'm not going to go into a deep dive on because it is actually very heavy math. Historically, the latch has been understood to just consist of the flexor muscles, the ones that are keeping it closed. They evolved a little lump in the tendon that basically goes over a lump inside the appendage and braces it, so some mechanical advantage. And for many, many, many years, we've understood that the flexor muscle relaxes, which then allows the system to rotate out. But for all these years, we've had two major conundrums. One is that's a terrible idea for the flexor muscle because there's no way that that latch could get out of the way in time for the whole system not to yank and break the flexor muscle. But secondly, we could see in our empirical data that the flexor muscle relaxes long before the appendage actually moves. So we just published a paper in PNAS, which I'm not going to get into today, that does the math showing that in fact these animals have geometric control of their latch. They actually do a funky thing with torque where they have to pass a point in order for the spring to actually push, a geometrical point for the spring to actually push and open the appendage. So it's not on here, but just as an aside, it's pretty cool and it's worth looking at if you're interested. Okay, flow. Last point on this slide, flow. Check this out. This is the duration of 300 milliseconds for the muscle contraction. Release of the latch, about 30 milliseconds. The spring pushes the appendage for 0.9 milliseconds. The appendage rotates towards the target in 1 millisecond. The hammer impacts the target in 49 microseconds. The impact is at such extraordinary speeds that it causes cavitation, which is essentially vaporization of the water. Cavitation is one of the most extreme energetic events, period. Physics, biology, whatever, you name it, because an enormous amount of energy, well, a small amount of energy is released over an exceedingly small period of time. It causes heat, light, and sound, and these animals basically conclude this strike with a collapse of the cavitation bubble. This right here is extraordinary morphological and mechanical control of energy flow. This control is what makes it so that these animals do not have to use gunpowder in a bullet and self-destruct. This, when we talk about morphology, biomechanics, and limits, and sort of extraordinary abilities, this is exquisite control of energy at each stage, compressing time, by starting with a small amount of energy, but releasing it over shorter and shorter and shorter periods of time, so that in the end, this animal is generating one of the most extraordinarily energetic events ever documented in terms of physics, which is cavitation. So amazing, right? I mean, I think that's pretty amazing and much more interesting, in my opinion, than being in the Guinness Book of World Records. Although I never complain about that. That's fine. But I think this is much more interesting from a limits, morphology, mechanics perspective for those of us who are into that. Okay. So when we talk about this exquisite control of energy, this is what it looks like in real life. This is kind of one of my more favorite graphs. So I feel like I show it in every single seminar for like the past 20 years or 15 years, whatever it's been. But I still really like it. Check this out. So if you use the Mantis Shrimp approach or any of these systems that low mass moving materials to generate fast motion, this is what they get. With a single appendage, they generate an impact and then a cavitation. And then with a second appendage, they generate impact and cavitation. This happens at microsecond time scales. This is a fairly wimpy peak force, so about 700, but they can get up to 1500 newtons. And compare this to another shell eater, which is a horn shark. Milliseconds, this is the best, by the way, peak force that they generate for the horn shark, and there's the shrimp strike. But when we talk about energy, this is a lot of energy under this curve, and this is extraordinarily little energy under this curve. And because these animals and other animals have evolved this ability through morphology and mechanics to exquisitely control energy flow, it means that the mantis shrimp strike is even with the peak force of a hyena. So essentially, this is an alternative ecological tactic or behavior, if you want to call it, for breaking hard-shelled prey for a very small animal with a small amount of energy that has evolved a way to control the rate and the flow. Just to hammer home this point, haha, one last time. This is the equivalent of energetic control of the difference between 1 year and 1 second, or the size of the planet and the size of you. So this is why some of us decide to devote our lives to studying mechanics and morphology, because that's kind of mind-blowing. Okay, so hopefully that gives you a sense so far of what I mean by amount, rate, and flow, small, springy, and ultrafast, some concept of the limits. But now let's look at interdisciplinary for a minute to wrap things up. So during all this sort of seismic realizations we've had, almost all of this has occurred in the context of a large multidisciplinary grant that I run that involves micro-roboticists, centimeter scale roboticists, material scientists, physicists, mathematicians, and biologists. I am a massive advocate of interdisciplinary work and an advocate of expertise. And I think this is forgotten sometimes, but for the biologists on this call, our abilities to work with organisms and discover things about them and quantify what they're doing is absolutely critical, and it's very difficult to do in order to populate and move forward an interdisciplinary field of these types of energy controlled motions. So I want to just advocate for that for the biologists here to understand the extent to which we bring extraordinary expertise to this space. So we've been working on this in a number of ways, but I wanted to tell you one last little story about trap-jaw ants. So this is, these are some physical models / robots made by Zeynep Temel, who's now at Carnegie Mellon, and she was at Rob Wood's lab at the time at Harvard. And we had this sort of funny exchange in this space of interdisciplinary things, where I basically was like, here are all the morphology papers on trap-jaw ants. This is how we understand their mechanics. So you could just go build something that works like that. And this set off what has been basically a 5-year journey, which I will tell you in less than 5 minutes, painful as that is, about how trap-jaw ants actually generate among the fastest repeated use movements on the planet. Trap-jaw ants generate extremely fast jaw rotations through LAMSA, latch mediated spring actuation. They push their jaws closed with a spring. The spring is their entire head. which changes shape and pushes the jaws closed. They can also close their jaws with the muscle directly. So this is a multi-purpose appendage, if you want to call it that, just like the mantis shrimp. Just like the mantis shrimp, they can move on either side of this transition. And that's a cool thing about biology. Usually biological systems don't throw out the baby with the bathwater. They can manipulate here and with multi-axis control through direct muscle contractions, and they can shift over into using a spring-based system in order to generate extremely high speed strikes. When we decided to measure the jaws closing, we discovered something kind of crazy, which is that the jaws close with a very precise circular motion. So if you track the tips of the jaws, they're perfectly circular. And it's so clean that if you do a derivative for velocity and acceleration, it's still clean, which for us, that's like unheard of in biology. So we started to wonder how that was possible, that they could produce such an extraordinarily precise motion over extremely short time periods. So what you can see here is the head changing shape, and as it changes shape, it's like puckering in on the sides. And when the pucker extends out, it pushes the jaw closed. And same here, puckered in, puckered out. And we figured all that out, that in fact it does cause all the displacements add up, so that the head is actually pushing the jaws closed. And yet the jaws themselves are very loosely articulated to the head in order to allow for multi-axis motion for slow manipulation with the muscles. So what we discovered is that these ants use what we term a dual spring force couple. When they're unloaded, they can be used for muscle actuation by just simply contracting a muscle here. When they're loading, they latch the system so the jaws can't close, and both the head capsule flexes to store elastic energy and the muscle apodeme unit stretch to store elastic energy. So we have a stretch and a compress, and the two of them together torque the jaws closed. So some of you might be like, okay, listen, I said I liked morphology and I like mechanics, but this is really over the top. My brain is dead. I don't want to talk about this anymore. Well, this might not interest you. This diagram might not interest you. But what might interest you is how this connects back to the theme of this talk. Small, springy, and ultra fast. It turns out that if you do the energy accounting of this jaw closing, you cannot get enough energy stored in the traditional tendon apodeme strap thing. You would have to stretch this piece longer than the head of the ant. You cannot pull it enough to get enough energy to fire these jaws. So what we're actually seeing here through this head compression, and this is exactly what the mantis shrimp are doing and so forth, is distributed elastic energy through three-dimensional morphological structures, where if you have a cylinder or a head capsule, you can change its shape by tiny amounts all through the three-dimensional structure in order to store that energy. This is really crucial, especially if we move back into understanding limits, multifunctionality, and robots, and this notion of morphology. When I put this up originally, and of course in our very diagrams, our notion of springs in engineering and in our heads relate to our tendons, our Achilles tendon, pogo sticks, whatever. But the truth is, these organisms are using three-dimensional geometries in order to distribute tiny displacements to store elastic energy. So when we talk about moving the capabilities of robots and the like into a space where they can be repeated use and driven by springs, we should really be thinking about morphology, and we should be thinking about three-dimensional structures and how they're loaded. Because once you move into that space, then we might be able to move more engineered systems in that space. But we also start to understand really the morphological and mechanical strategies to allow organisms to move over into this space. And it was really through this kind of crazy journey with the trapjaw ants that we finally realized this dual spring force couple, but also figured it out in the mantis shrimp as well. And it turns out it's relevant for locusts. So it's a broadly relevant framework. Okay, so to wrap it up, I want to just talk about this thing that I mentioned at the beginning, because I know Martha's here, and I know some of you really love morphology. So when we talk about laws, if we want to talk about a law, I'm not brave enough to say that I have a law here, but let's just say I believe we have identified a major transition in movement that's relevant to biology. So as a comparative biologist, it starts to be very interesting to see how animals navigate that from the perspective of size. So one of the projects we've been working on for a while, we have an NSF grant right now to do that, is to look at how mantis shrimp traverse this size range, which they in fact do, during growth and development, but also across evolutionary timescales. And to understand how organisms and evolutionary processes navigate what appears to be a major transition and how to generate motion. That's a picture of a larval mantis shrimp over here. And then the other piece that we're now finally working on, and actually just finished, which is incredibly difficult, talk about a math project, is to move forward from the notion that these ultra-fast animals are just about being the fastest animal on the planet or the fastest organism on the planet, and instead think about their controllability in different environments, how they can adjust their inputs and their outputs. And we have found that they in fact can through spring and latch processes. And this, from an ecology and an evolution and behavior point of view, for me is probably the most exciting thing, because now we don't have to talk just about extremes. We can talk about the whole space of evolution and trade-offs and behavior in these systems, and that they can be doing things like adjusting through development, evolution, or behavior, how they operate their latches and springs. That may seem obvious to you right now, but that is complete 180 from the paradigm for the system today, which is that they do not have any control once the motion has started. So with that, I'm going to thank you all for your attention. I hope that was a fun kind of set of themes to run through. And I wanted to end on just a little plug. I had talked about the challenges of being thrown on and off planet science by COVID, but I also want to acknowledge that there are a lot of folks out there who want to be part of planet science and don't have the doors open to them. And I wanted to invite you to check out a project that I've been working on for 10 years now, which is to enhance equity, accessibility, and transparency through a software app that I developed that we run now at multiple universities that allow anybody to see opportunities for research and not just have to stand back here. Because for me, as I've been off this planet science, I've missed it. But I also always want to invite more folks on. Huge number of people to thank. I cannot even begin to tell you, as I hope you can imagine, what I just told you was literally probably 100 people's worth of effort. Everything from my kids on up to the Muri team, which we finally got together after 2 years recently, the Duke Maureen Lab, and again, this interdisciplinary team, many of whom are named here, but especially the Azizi Lab, the Bird Brighter Lab, the Crosby Lab, Ilton Lab, and the Wood Lab have been primarily involved in all this. All right, I hope there's some time for questions. I'm also just happy to do whatever you all like to do, but I appreciate your attention, and I hope that touched on a few of your topics for your group.
Wow, thank you so much, Sheila. That was a fantastic talk. Really
Thanks.
a lot of food for thought. Does anyone have any questions?
Kathleen, do you want to call them out? Okay, cool.
Hi, Sheila. I'm not sure my video is working. Sorry for that. I really enjoyed your talk. Thank you very much for that really cool introduction to the biomechanics of animal movement. And I was wondering if some of the movement that you see in the plant world might similar to this LAMS mechanism that you were talking about. I'm thinking specifically about carnivorous plants.
Yes. Okay. So I left out like 10 million things. One really big thing I left out is that we do have a plant biomechanics LAMSA program in my lab run by Justin Jorge. And he works on ballistic plants. And it's actually really funny you bring that up because yesterday we were in here talking about that control theory paper that I put a couple images from. And we decided that plants were actually the best system to work on. Forget animals. But the reason why plants are so interesting in this space, and I think underappreciated, although plant people know all of this stuff, I guess it's underappreciated by the rest of the biologists who ought to know about it, is plants have extraordinary real-time tunability of their materials and the mechanics so that you can have something that is dehydrating or changing in the presence of different ions or amount of turgor pressure. And so when we talk about, there are many plants that are right up there as like the fastest, highest power output organisms on the planet. But they have this added ability to be very tightly coupled to their ambient environment. So stay tuned for Justin's work. He's finishing up a paper right now, but yeah, I don't know if you want to throw your carnivorous plant example. I don't know if you know that, but our Venus flytraps are native to North Carolina, so we're quite proud of those. But I don't know if you want to throw in some other examples that interest you that you were thinking of.
No, those are the ones I'm thinking about. Snap traps in Dionea or many other groups. I really look forward to that paper. Thank you.
Yeah, absolutely. Great question.
Yes.
Hi, I was wondering about the Amphipod claw closing. It's so small and it's so the acceleration is so high. Is that fast enough to cause shear cavitation like a snapping shrimp's claw? And does it want to do it for the same purpose? Or
Okay, great question. Yeah, I left out snapping shrimp. Two of my graduate students have been working on snapping shrimp. And if you don't know, snapping shrimp are different than mantis shrimp. Snapping shrimp shoot a water jet that forms a cavitation bubble behind the water jet, and then the cavitation bubble implodes, sorry, and generates a huge amount of noise and knocks out their prey and so forth. And they also use latch mediated spring actuation. Stay tuned on that paper. I was working on that this morning. Their spring mechanism hadn't been entirely worked out. We finally got that done. But the amphipods, okay. I love the amphipods. So the amphipods rarely cavitate. And this is going to get me into a space where I'm like, if anybody on this call wants to start working on this, like, come talk to me because it's so hard. The mantis shrimp and the amphipod both move faster than what engineers and physicists call the cavitation threshold. In other words, there's a threshold above which the speed above which cavitation forms. And so this is like the speed limit for fish, fast swimming fish go underneath that speed limit. All kinds of like missiles and things like that. Whenever the Navy's doing something underwater, they're often considered thinking about the speed limit because once you go above the speed limit, you make a huge amount of noise. So both of these organisms are going faster than the speed limit and not cavitating during the forward motion. So one of the things that they're doing, which I have never been able to explain because the visualization is so difficult, is that both of these animals have fairly loosely articulated dactyls. So they're actually quite wobbly. And they are all, I always show it like, like a sort of 1 degree of freedom chop type motion. But in fact, they are highly curved and asymmetric, and they basically slice through the water. And if you looked at the micro CT scans of the amphipod, you'll see it's like this gorgeous blade that essentially slices through the water. In the mantis shrimp, if I attach like even a 1/10 of a millimeter diameter bead, which we would love to do for visualization, it causes like catastrophic cavitation as it slides through the water. So that was really not what you were asking. You were asking a yes or no question, but it happens to tap into something that I'm extremely interested in that is on the topic of limits of visualization. You basically have to figure out how to do 2D PIV at 500,000 frames per second in a fluid that's at the verge of cavitating. Anyway, it's very, very difficult to do.
Well, if it's about the onset of cavitation, it might also have to do with nucleation of the vapor bubbles, because as your scale gets smaller and your time scales get smaller, there's time needed for these things to grow before everything has happened. So.
Yes. Oh, I love that topic. I won't go on a big deep dive, but the times, you're absolutely right to be thinking about time scales, because once you're talking about things that are so brief, the encounters with like turbulence or seeding bubbles or whatever that, that are the source of the cavitation, again, shifts way out of our intuition. So it could, you're right, it could just be probabilistic, which would not be as exciting as if they have some very cool fluid dynamic way to circumvent
I would be pretty excited about it. I have a lot of experience with cavitation and very little with biological systems. So, yeah.
Awesome.
Thank you.
Thank you.
Other questions?
I have a question if no one else wants to jump in. And I was really interested, especially with that last bit where you were talking about using three-dimensional surfaces to store elastic energy and thinking about, morphology as we often do. And whether or not you're going down the path of modeling even to see how the morphology of that surface impacts the amount of elastic energy that can be stored, and whether or not we see differences in the shapes of these structures to allow, either like more fine control over how much gets stored, or minimal deformation stores maximal amount of energy, like that kind of thing.
What a great question. So what you just asked is essentially my question. And it's a really cool intersection, which I feel like folks in applied math have been working with for quite a while in terms of thinking about the mathematics of shape and how you can have a small change here that that causes a bigger change, or different types of changes of a 3D structure. I actually think this is precisely in that space. And when you get in that space, then, as you very astutely have observed, you start to have a pretty cool parameter space, because you have, in these systems, you have how much energy you can store in the structure as a whole, but you also have to attend to where and how is work being delivered to the moving object. Those are two separate problems, because you can put all kinds of energy in a structure and then release it and not move those trap jaws anywhere. But you really have to be able to actually have the geometry of energy delivery tied into the overall morphology of the elastic energy storage structure. Again, I got more than enough to do. So if any of this interests anybody, talk to me, because I really love this topic. And actually, it's an area of math and modeling that I've never worked in terms of these structures. And this is, it's a rich space in applied math. So I think it would be so exciting to bring some of those principles over to biology. And now we have some systems like the trap jaw, where we finally have actually, frankly, formulated the problem. We had been conflating for so long energy storage and energy delivery, and it doesn't make sense from that perspective. But if you separate them out and ask, how is the head an energy storage device? And then how does the head deform in order to geometrically deliver work in a very precise way, then it's a far more clear problem. Some of you might be like, yeah, duh. Well, say yeah, duh to me after you bang your head against something like this for 5 years. When you know, at the end, I guess what I'm going to say is like, maybe what I'll say is this: my best discoveries in science have all been endless amounts of work, at the end of which I look back and I'm like, oh, dang, I have my high school college physics book underneath my computer here. I'm like, it was there the whole time. I just couldn't see it. Mimi Cole has a great way of like, she's one of the greats in comparative biomechanics. She has a great line for that. It's something like, it might be there the whole time, but you have to do the work in order to actually see how these fundamental principles of physics emerge in these biological systems. And it's just not obvious, even though it is at the end. And when you show it to people. Anyway, ramble.
Okay, so Travis and Marta, I'm not sure who is first.
Sorry, were you saying that I'm able to share my question?
Yes,
Oh,
please.
wonderful. Thank you. Sheila, that was so great. Thank you so much. I really enjoyed all of the work that's happened in the past few years. This has been a great catch up. You got me thinking a little bit, and I wanted, and in particular, I was really struck, of course, with this inflection point between ballistic and muscle-mediated movement. And the x-axis, as I recall of that plot, was accelerated mass. So this got me thinking, are there larger organisms that contain smaller ballistic structures within them that they use? And do these follow the same principles, the same inflection point, or are they fundamentally different in some way?
Oh, Martha, that's awesome. And I'm actually really glad that I'm very honored you found time to watch this talk, but it is an update on what we've been doing since you left. So that's a really good point. And again, with this notion of like kind of obvious in hindsight, but took us a while to get there, those two words on that x-axis are all, it's like, that's the name of the game. An organism can be huge and be operating a tiny structure using elastic mechanisms. So you're right that the accelerated mass is just the mass that's being moved. And again, it's kind of funny, but I literally remember the moment on that science paper, the Ilton et al. Paper that I brought up earlier, where we defined that transition point, where someone on the team, I believe it was Dr. Suzanne Cox, she's like, you're missing the whole part about the arm not throwing the arrow. So the thing, when we talk about small energy and the utility of springs, you have to get the mass off of the system. This is a huge limit. So that's like probably 3/4 of the magic of archery is getting your arm's mass off of the thing you're accelerating. So yes, it's all about accelerated mass. So when we think about evolutionary transitions and how organisms navigate this mass transition, without question, you have to consider how they've evolved to take the weight out of it in order to be able to use a spring to do so. So it's a pretty rich space for morphology. But for the most part, and by the way, just as an aside, this is super relevant for jumping insects. So like lots of locust work has been studied, like the whole body of the locust jumping. So that's where it has to accelerate its whole mass. But they also can kick with their leg. And of course, that has very little mass. And likewise, I didn't even put this on there, but the trap jaw ants, they fire their jaws closed to capture prey, but some have evolved the ability to jump with their jaws. So they're basically jumping with a higher mass. So even within the use of a system, they may be experiencing totally different mass loading. For the ants, they're always to the left of that mass transition line, even when they're jumping their body. But other organisms, it matters a great deal. Great question.
Thank you.
It's Travis' turn now.
Maybe he was just showing his video. I don't know.
No, yeah. No, I think this is all super, super cool stuff.
Thank you, Travis.
I wanted to throw an idea out after Kathleen's comment. So when you started talking about deformation of the whole shell, the whole ecosystem, the whole body shape, right? The whole exoskeleton. The first thing that jumped into my head was unibody cars, right? Because engineers build cars to deform and absorb a ton of energy. So is there any optimization that engineers have put into crash crumple zones, controlling how and where that energy goes into a whole structure, a whole shell, to absorb energy? There's got to be something you could steal from that, right?
Yeah. Okay. So Travis, this is a really good question, and I don't know that if you know me well enough to know this, but like, this is the kind of thing that I'll spend like a month reading about in like engineering textbooks and literature, where I'm like, well, have they dealt with this yet? Have they dealt with this yet? So I have two answers to that. The first one is that engineers, and of course, this is a gross generalization, but it's not incorrect. When engineers use springs, they're typically talking about dissipation. So what you're talking about with like basically, you want your car to crumple because that's going to take a lot of the energy out of the crash. And then all kinds of other kinds of dissipation. And then resonance and oscillation, so efficiency. Vibrating systems and things like that. engineers really don't do a lot with what I'll call spring propulsion. So, when are you actually going to push something with the spring? Now, there are lots of examples of it, but like when you look at like engineering design today, that doesn't come up very often. But when we go, and this took me forever to understand, when we go into car design space, which I have spent a ridiculous amount of time on, it is the wrong scale. So the energetics of spring propulsion require, for it to even be worth considering, it has to be incredibly small. So does that mean that three-dimensional car design is irrelevant to spring propulsion? I don't know. But my bigger problem is that this hasn't been figured out by the engineers, or at least not published in a way that I can find. So I can't tell whether you can go from like a big 3D, crash type thing, which has been studied a ton, to a principle of a tiny structure storing energy. So generally speaking, my better luck has been working with materials scientists who do deal with this a bit at pretty small scales. They're kind of hyper-focused these days on like nano scale energy storage and release. I'm having better luck there, but I totally know where you're coming from. And it's a really good question.
Right. And I think the analogy would only work so far, because then the next thing you said was you can dump a bunch of energy into a system and distort it, but you need to be able to get it back out in an organized way.
Yeah.
And we crash a car and it's all smashed and we don't need to get that energy back. So it's only going to work. So the comparison's only going to work so far.
Yeah, but I still think whenever we can follow some, places where a ton of work has been done, there probably are some nuggets in there. And I kind of think that trap giant head looks like a car's, sort of like, a three-dimensional squishy thing. Anyway, I love it. Great ideas, great questions.
Great. So I think maybe I'll stop the recording at least. Thanks everyone and
Yeah,
Sheila,
thank you.
thanks so much for that fantastic talk. Stimulated a lot of discussion, which has been fantastic.
Thanks all of you. I really appreciate it.